after transcription, you have your messenger RNA molecule that is now ready to be converted into a protein. So your messenger RNA molecule is translated into a protein using the genetic code. So the genetic code basically says that for every 3 nucleotides within your messenger RNA molecule, those 3 nucleotides are going to determine which amino acid is going to get added whenever you make a protein. So these triplets are called codons. So if you hear the term codon, it's referring to a sequence of 3 particular nucleotides in your messenger RNA. So we could say that these codons are in your messenger RNA molecules. And in order to be able to go from your messenger RNA to a protein, you need to have a chart. So that chart is something that's given to you on a test, and it looks something like this. So in this chart, basically, if you look, it says first base here. So we're not playing baseball or softball, but first base, second base, third base. First base is either going to be a U, C, A, or G. So in that particular codon, you find wherever your first base is. So let's say you had C, A, U. The question is what amino acid would get added? Well, you find C as your first base, the second base is A, and then you come down to where the third base over here is U, and you would have CAU, and that would tell you kind of next to it on the right here is a histidine. So we would add a histidine amino acid. So again, the top part here would be your codon that's in your messenger RNA. His would be the amino acid that gets added to your growing protein. All right, so in translation, messenger RNA has a sequence of codons that basically determine the order of the amino acids. Those codons are non-overlapping. So in other words, if you had C, A, U, G, C, U, A, C, like that. In those 9, basically you have individual sets of 3. So you would have, in this particular case, this would be your first codon, CAA, then UGC, then UAC. Right? So you move from set of 3 to the next set of 3. Whenever you actually go from the codon to your individual amino acid, that is done by these transfer RNAs that we've talked about in a previous lecture. Right? The transfer RNAs bring in your specific amino acids to the peptide chain. So if you remember, the tRNAs are the ones that have like that clover leaf kind of structure to it. The anticodon, which is what you can see down here, binds to the messenger RNA through complementary base pairing, and that's what brings in this new amino acid. All of this occurs on ribosomes, so this ribosomal RNA contains the binding sites where protein synthesis occurs. So all of these things work together. So kind of think of it as like cooking, right? The kitchen is where all the cooking occurs. That's your ribosome. The chefs kind of use specific ingredients to make a meal. So those specific ingredients, right, the flour and the sugar and the chocolate chips, because in my mind we're always making chocolate chip cookies, are the individual transfer RNAs. And they basically read the recipe and bring in the specific amino acids to make your overall product, which is going to be, after you get a whole lot of amino acids, you would have a protein. Right, so the protein is going to be the overall end product for that. Okay, so let's look at translation in a little bit more detail. So again, the first part of this slide just kind of shows you a few of the codons. It tells you the tRNA anticodon, right? So that UGU means that if you look here, well, I should probably go to a different color so it shows up a little bit better. UGU would bind through complementary base pairs to that. The anticodon would bind to the codon through the complementary base pairs, and the tRNA is what brings in your specific amino acid. All right. Now, whenever you're undergoing translation, there are 3 main parts. There's initiation, which starts the whole thing, then elongation, and then termination. So what do we mean by these? So initiation starts when your messenger RNA binds to the ribosome. So if you look here, this kind of grayish purplish outline here, that's your overall ribosome. So that's going to be the area where the protein synthesis occurs. The messenger RNA is kind of in the peach color here. So AUG is going to be a codon on your messenger RNA that always signifies to start translation. So the first codon is always going to be AUG. That's commonly referred to as the start codon. So the start codon basically says, okay, let's start protein synthesis. Let's start making a protein. So at that point is where you're going to start the whole thing. That start codon is always going to be AUG, which means we're always going to add a methionine to start things. All right, so that's going to be the initiation, right? Initiation starts with the AUG. After that, elongation occurs, and this is where we go kind of these 3 nucleotides at a time. So each one of these is a codon, so codon, then another codon, and we'll keep going all the way down. And what happens is when the tRNAs come in, the tRNA binds to the messenger RNA, right, through the codon, anticodon hydrogen bonding. That brings in a new amino acid. These amino acids end up forming a peptide bond, right? So the peptide bond occurs between those amino acids to basically continue to grow the protein. And that elongation process continues over and over and over and over again until you reach what's called a stop codon. So if we go back to the genetic code chart real quick, let's go back. On the genetic code chart, you'll notice there are 3 codons here that say stop. All right, so all of those say stop. What that means is you don't add an amino acid called stop. It means that you stop protein synthesis. So these ones are called the stop codon. And now that I'm back here on this genetic code chart, the other thing I wanted to point out is that our genetic code is redundant, which means that we have 20 amino acids that we need to add, and there's a lot more than 20 spots on this table, right? Because since things work in sets of 4, right? If you have 3 bases, can be 4 different things, the next one can be 4 different things, and the next one can be 4 different things. So if you multiply 4 by 4 by 4, you get 64 different spots on this chart. And for instance, if you look down here, you'll notice that all of these all make glycine. So typically, the last spot in your codon, right? So GG, and it doesn't matter what the last one is, GGG, GGA, GGC, or GGU, all of them make glycine. So we call this genetic code redundant because there are often multiple ways to make a particular amino acid. Leucine over here has 6, for instance. Some of them only have 1, like the methionine that we just talked about as a start codon that makes that adds the methionine, the AUG that's down here. That's the only way to make methionine. So again, you don't need to know all that. It's just kind of just for your knowledge moving forward because you'll always have this genetic code chart present for you. Okay. So, whoops. In other words, so we keep doing our translation until we reach that stop codon, UAA, UAG, or UGA, and at that point, the protein is released from the ribosome and is now going to be a completed protein. Notice all of these amino acids are linked together now through peptide bonds, and that protein will go do whatever it's meant to do. Like if it's an enzyme, it can go do its enzymatic function. All right, so that is protein synthesis. There's initiation at the start codon, elongation where you form all of the peptide bonds between your amino acids, and then termination, which stops the whole process and releases the protein from the ribosome. So having gone through all the information so far, you should be able to look at a problem like this, and if I give you the DNA informational strand, which is shown here, you should be able to come up with the template strand, the messenger RNA, and the resulting polypeptide that comes from it. And actually, for that matter, if I were to give you the template, you should be able to figure out the informational strand, the messenger RNA, and the polypeptide. If I were to give you the messenger RNA, you should be able to figure out the informational strand, the template strand, and the polypeptide. The only one you can't really do is go from a polypeptide all the way back through the other ones, because particular amino acids, because of the redundancy of the genetic code, right? There's one that, like if I gave you, for instance, said leucine, there are multiple options for what leucine could be in the messenger RNA strand. All right, so saying all of that, let's kind of go through this example and look at what would form from this particular DNA informational strand. So notice that we have the 5' end of the DNA molecule here, and we have the 3' end over here on the other side, right? So what we're going to do then from that, we're going to be able to write the DNA template strand. And the DNA template strand is going to go in the opposite direction. So instead of 5' to 3', it's going to go 3' to 5'. And it's going to be the complement. So T goes to A goes to T, C goes to G, and then A to T's, and so on and so forth. So you can go through and write the same T, C, A, 5'. So you're going to write the template strand, which is going to be the complement for everyone here. So T goes to A goes to T, C goes to G, and you do that across the whole strand. All right, so that would be your double-stranded DNA molecule, right? Those two strands that are up there would be found in that double helix together, held together by the hydrogen bonds between the complementary base pairs. Okay, so next up, we need to get to our messenger RNA. So the messenger RNA is going to be 5' to 3', and it is made from the template strand. So the A here, the complement is going to be found on the messenger RNA. So that A, the complement, you would think it would be a T if it was DNA, but remember in messenger RNA we don't have any T, so that's going to be a U. And then T is going to go to an A, G to C, and then we can continue going through the rest of the messenger RNA as follows: C, C, U, A, G, U. All right, so that would be our messenger RNA. So a couple things about that messenger RNA. Notice it's going to be the complement of the template. And anytime you make something that is a complement of something, you always have to change. You go from 3' here to 5' there. So you're always synthesizing new molecules in a 5' to 3' direction, and you synthesize them as the complement of the previous molecule. So the one thing that you should notice from this is that the messenger RNA strand is basically identical to the DNA informational strand, except T goes to U. So in other words, the DNA informational strand and the messenger RNA are going to be identical, except in messenger RNA we're going to substitute U's anywhere there was a T. Other than that, it's going to read the same. So that's why this one, it's going to be CCU instead of CCT whenever we look at the messenger RNA. Down here, right? AG, instead of AGT, I'm going to put AGU. All right, so now once you have the messenger RNA, then we can go and make the polypeptide. Now, for the polypeptide, remember for proteins, we don't have a 5' and a 3' end. For proteins or polypeptides, we have an N terminus and we have a C terminus, right? Again, different biological molecules, they have different property. Remember the 5' here means you have a 5' phosphate on your messenger RNA or on your DNA, and on the 3' end you have a hydroxyl group. For your polypeptide, the N terminus means you have a free NH3+, and on the C terminus you have a COO- over there. All right, so again, remember those key features of these various molecules. All right, so now in order to go from the messenger RNA to the polypeptide, we need a genetic code. So our genetic code is going to tell us what the various codons, and again, the codons are the sets of 3 here, what they encode for in terms of the protein. To go from UAC to polypeptide, we need our genetic code chart. So I'm going to go back a couple slides, and we can see that the UAC, whoops, one too far, U is here, then A, and then down to C is going to be our tyrosine, is going to be the first one. So the first here is going to be TYR. AAC, I'll do one more and then I'll just write the rest of them in and trust you guys can figure it out from the code. A, you start here, then you go to your next one, second base is A up there, your C over there, where do they all go? You go here to there, down to this one. AAC is going to be ASN, or asparagine. So tyrosine, then ASN is asparagine. CCU is going to be proline. CGG is going to be arginine. CCU, again, is going to be proline. And then AGU is going to be serine. So the resulting polypeptide would be tyrosine on the N-terminus, then asparagine, proline, arginine, proline, and then serine on the C-terminus. So again, you should be able to do this whether from a DNA informational strand, you should be able to do it if I give you a template strand, or if I gave you the messenger RNA. If I gave you just the messenger RNA, you should be able to come up with your 2 DNA strands as well as the polypeptide.